Hey everyone, and welcome! Today, we're diving into a fascinating story of friendly fire happening right inside our own bodies. It's called type 2 hypersensitivity, and it's all about what happens when our immune system's most precise weapons, our antibodies, make a huge mistake and start attacking our own healthy cells. It's a wild ride, so let's get right into it. So, right off the bat, here's the central mystery we're going to solve today. How can the exact same kind of immune screw-up lead to two completely different results? You know, on one hand, it can be a total wrecking ball, just demolishing cells. But on the other, it can be this subtle saboteur, hijacking a system without breaking a single thing. How is that possible? Well, that's the puzzle we're here to unpack. Okay, so what's actually going on here? At its heart, the problem starts with two specific kinds of antibodies. You've got your IgG and your IgM. For some reason, they get confused. They see a molecule, an antigen, on one of our cells, and they decide it's an enemy, and they lock on. That one binding event, that's the trigger that kicks off all of the damage. It is a classic case of mistaken identity. And believe me, the consequences can be serious.
Now, all the chaos that follows falls into two big buckets. The first one is pretty straightforward: cell destruction. The antibody marks the cell, and the immune system just completely eliminates it. Gone. The second path is way more subtle: cell dysfunction. Here, the cell isn't killed, but its entire function gets messed with, sometimes in a really dangerous way. Let's start with the brute force approach first: destruction.
All right, path number one, direct destruction. You can really think of this as the immune system sending in the wrecking crew. There's no subtlety here. A decision has been made that a cell is an enemy, and it needs to be taken out completely.
And here's the perfect way to picture it. The antibody is basically just slapping a giant target sign onto one of our cells. Once that sign is there, other parts of the immune system, the authorities in this analogy, see it, and their only job is to destroy whatever has that sign on it. In this case, it's our own perfectly healthy cell.
So who exactly are these authorities that come in to do the crushing? Well, they come in two main forms. First, you've got the complement system, which is this cascade of proteins that just unleashes total chaos. The second is a much more direct attack called Antibody-Dependent Cellular Cytotoxicity, or ADCC for short. And this one involves bringing in the special forces. So how does this complement attack actually work? It's a really powerful chain reaction. It all starts when that antibody binds to the cell. That acts like a tripwire, activating a whole series of proteins floating around in your blood, called the complement system. And once those proteins wake up, they get to work, and the end result is the complete destruction of that cell. It's incredibly fast and devastatingly effective. And this complement system has a whole arsenal of weapons. It can form something called a membrane attack complex, which literally punches holes in the cell membrane until it bursts. It can also release signals that scream for backup, triggering massive inflammation and calling even more immune cells to the fight. And finally, it can opsonize the cell, which is a fancy way of saying it coats the cell in signals that basically make it look irresistible to other cells called phagocytes, whose job is to eat things. Now, that second method, ADCC. This is less like a riot and more like a targeted hit. Again, the antibody sticks to the target cell, but instead of triggering that complement cascade, it acts like a beacon for specialized assassins, particularly a cell called the natural killer cell, or NK cell. The NK cell comes in, grabs onto the antibody, and then injects a lethal cocktail of chemicals right into the target, killing it up close and personal.
Okay, let's switch gears. We're leaving the wrecking crew behind, and we're entering the world of espionage. This is path 2. The goal here isn't to destroy the cell at all. It's to hijack its function from the inside, causing chaos in a much, much quieter way.
And the traffic light analogy is just perfect for this. The cell, our traffic light, isn't getting smashed to pieces. It's still there, it's still standing. But a hacker, that's our rogue antibody, has taken control of the signal. They can force the light to stay permanently green or jam it on permanent red, snarling up the entire system without leaving a scratch on the light itself. The scientific term for this kind of sabotage is antibody-dependent cellular dysfunction. And the key here is that the antibodies aren't just binding anywhere. They're specifically targeting the receptors on the cell's surface. You can think of these receptors as the cell's control panel. By latching onto them, the antibody can either jam the controls or hotwire the whole system. So let's look at that stuck-on green scenario. A classic example is Graves' disease. Here, antibodies bind to a receptor on thyroid cells that's supposed to wait for a specific hormone to turn on. But these antibodies, they mimic that hormone perfectly. They essentially get the key stuck in the ignition, jamming the on switch.
This forces the thyroid to pump out hormones 24/7, leading to hyperthyroidism.
And what about stuck on red? Well, for that, look no further than Myasthenia Gravis. In this case, antibodies target the receptors on our muscle cells that are waiting to receive a signal from our nerves. By binding to these receptors, the antibodies create a physical roadblock. The nerve is sending the signal to move, but it just can't get through, and the result is debilitating muscle weakness.
So, we've got the two main paths down: total destruction or subtle dysfunction. Now let's just quickly connect these ideas to a few more real-world examples, just to see how massive the consequences of this one immune error can be. This table really lays it all out beautifully. You see a transfusion reaction when someone gets the wrong blood type? That's a classic example of destruction. Your antibodies are targeting those foreign red blood cells and calling in the wrecking crew. Some types of anemia caused by drugs, same deal, destruction. And then you've got Graves' disease and myasthenia gravis, are star examples of dysfunction, one by overactivation, one by blockage. It's amazing, right? Different targets, totally different outcomes, but it all starts with the same initial mistake. So after all that, what's the big takeaway here? What's the one thing you should remember? Well, it all comes down to really understanding that core difference between these two paths. When it's destruction, the antibody is basically a death warrant. It marks a cell, and the system physically removes it. End of story. But with dysfunction, the antibody is a hacker. It doesn't destroy the cell. It just gets into the control panel and messes with how it works. It really all comes back to that central question. Is the immune system crushing the car, or is it just hacking the traffic light? And that leaves us with a pretty powerful thought to end on. Our antibodies are supposed to be our most precise, highly trained defenders. But as we've just seen, they can also become our most specific and dangerous saboteurs, capable of either brute force demolition or subtle systemic sabotage. It's just a stunning reminder of how complex and sometimes how fallible our own immune system truly is. If you found this journey into the immune system as fascinating as I did, then please go ahead and like this explainer and be sure to subscribe for more deep dives. Thanks so much for joining me.